Hey guys, we are looking at section 12.2 for today. And this one, the title for 12.2, as you can see, is Applying Mendel's Principles. So we're going to kind of build on what was done in 12.1 and start looking at a little bit more detail in terms of, how we can track inheritance and also predict the inheritance of specific traits in offspring when we have parents of certain types. So with this comes Punnett squares. And I think what I'm going to do is do Punnett squares as a separate screencast. So for this section, what I'll do is basically go through the section notes with you, including some of the Punnett square stuff with a focus on vocabulary. All right. And then in terms of like actually walking you through how we should set up Punnett squares, I'll have that in a second screencast. Okay. So just so we're not, confused about where to find that information. So as always, go ahead and get the title for me on this one. And so what we're going to look at again is really tracking traits within a family, right? How do we predict potential offspring traits? And we're going to talk about these things called genotypes and phenotypes, which if you watched the video from last week with Mendel's P's, they use those terms in that video. They also use terms like dominant and recessive alleles, and all of that is going to be defined for us here in this section. All right. So we talked about in Mendel's time, right? When he did his research, one of the things we mentioned is that he had this idea, and it was pretty novel at the time, but it was an idea that traits are inherited and passed down as discrete units. They are not simply parent traits kind of blended. And so as discrete units, we've now learned to call these things genes. Genes are the name that we give to those discrete units that he once saw being passed from generation to generation of pea plants. And we know genes now are what we hold within our chromosomes, right? So genes have specific sequences of DNA, specific codes, and those codes specify specific traits, right? And traits are kind of what we see on the outside. Genes are what we hold on the inside of our chromosomes, right? It's the actual DNA you got from mom and dad. So the way that we see a trait being expressed, the way that we can actually see a gene physically in the appearance of someone is in the production of proteins. So the second bullet here is super important. Genes, the real purpose is to provide a set of instruction that allows a cell to make a specific protein. And different genes code for or can produce different types of proteins. It's the combination of the proteins that are expressed that determine what we actually look like. So brown hair is a specific combinations of genes that are used to make a specific combination of proteins. And as those are produced, that's where we start to develop this, what we call phenotype. And we'll talk about that here in just a second. So when we look at specific types of genes, we use this term allele to represent those different variations of a gene. So within this allele category, we can have alleles that we consider dominant, and we have alleles that we consider recessive. All right, and we'll get to that here on the next slide. But in this specific example, Mendel's research tells us that the round shape here is actually considered dominant. The wrinkled shape is actually considered recessive. The yellow pea color is actually considered dominant. And if this was green, which it's not, would be considered recessive. So characteristics, we know, like physical characteristics, are what we determine based on the genes that we inherit in our chromosomes from mom and dad. The differences in those characteristics are determined based on the different varieties of genes that we can actually obtain. And those different variations are what we call alleles, okay? The allele in this case, like I said, for yellow is going to be dominant. And this dominant should be up higher. I'm realizing that right now. The allele for yellow seeds is dominant to the allele for green seeds. So that dominant word needs to be scooted up a little bit if you're jotting that down. But yellow is the dominant color, and what that tends to mean is that it's not necessarily that it's more powerful, but you can maybe think about it that way. When we say dominant, really what we're referring to is this idea that you don't need quite as many copies of a dominant allele to see that dominant trait. All right, so let's look at what that actually means. So in looking at dominant versus recessive types of alleles and then traits, remember, we know parents each donate only one copy of every chromosome, one copy of each homologous pair, or 23 total. And each gene is found on a different chromosome. And parents, again, only donate one of each type of chromosome, right? So That essentially tells us that each parent only donates one allele per gene, okay? So when we think about it that way, that means mom can either donate a dominant allele or mom could donate potentially a recessive allele. It kind of depends on what she has, right, and what she's able to give. Dominant alleles in general, when we write them, are represented by capitalized letters. And I usually, when we do a Punnett square, and you'll hear this in that screencast, but when I do a Punnett square, I call those dominant alleles, I say big something. So big A, big B, if it's a capital B, big C, whatever letter it is, instead of saying capital, because it's just easier to say big. So if you hear that, that's what I'm referring to. Recessive alleles are represented by lowercase letters to represent the fact that they are kind of covered up maybe by those larger, more dominant alleles. And so for these, instead of saying lowercase, I usually say little. So like little a, little b, little c. When we go to actually do those, I'll explain that a little bit more, okay? But in terms of dominant alleles, what this tells us, we know that dominant alleles get expressed if an individual has at least one copy present. That's all that you need. So purple flowers are dominant for pea plants. And we know that in this case, if a flower, a pea plant, we know if a pea plant, the seed that was created and from which it grew, if that harbored two dominant alleles, big P, to represent purple and purple, then that means that my phenotype, what I actually see for my trait, is going to also be purple. So dominant plus dominant creates a dominant trait. Now, this one is kind of the unique scenario, right? We have a dominant allele, big P. We have a recessive allele here, which causes a white flower color. But when these two combine, we don't mix those colors. All that we see is whatever's dominant. So the dominant trait here, the purple, is still the one that we see on the outside. It's the one that codes for that flower color. It's a protein that's made. And so what this means is that if you look at a pea plant that has purple flowers, well, we can't always identify exactly what the genes are that are found within those cells, because it could be that they have two dominant alleles, or it could be that they have one dominant and one recessive. The only combination that we can truly identify is when we see a recessive trait, and that recessive trait indicates that there have to be two recessive copies, two recessive alleles here. Because anytime a recessive is paired with anything else, it simply won't show. So these two items here are going to be important. Dominant is expressed and shown when there's at least one dominant. All you need is one. But recessive only shows up when you have two, okay? So we just talked about the fact that parents only donate one allele for each gene because they only donate one chromosome per homologous pair. And for those alleles, if we have two that are exactly the same, let's say mom and dad actually did pass down the exact same copy of that allele, which would be this scenario here, we call that homozygous, because homo meaning same. These are the same alleles, okay? Heterozygous, on the other hand, is where mom and dad donate two different copies of alleles. So in this case, round is dominant to wrinkled. And in this scenario where something's heterozygous, dad maybe here gave the round, the big R allele, and mom here maybe gave the wrinkled, the recessive allele, okay? And so what we see again, if you think back to what I just said on the last slide, as long as you've got one copy of a dominant allele, that's what you get to show. And so in this scenario, I have a round, and I'm going to show that round trait. I am not, however, going to show the wrinkled, but because this plant inherited both of these from mom and dad, this plant could pass down this one to its offspring or the round one to its offspring. So heterozygous does matter when we talk about reproduction. It's just that sometimes it's hard for us to identify it based on just, physical appearance. So I've used these words a little bit already, just because I get in the habit of using them, and they're words that we use often when we talk about genetics. But genotype and phenotype, you absolutely should know the difference between these. Genotype refers to the genes found in any organism, in any cell. It is like on a Punnett square, it is actually the letters that you write down, okay? Not letters that spell out a word, but just the symbols like big A, little a. Or like on this slide, big G, or big G, or little g, little g. Those are all what we call genotypes. Phenotype, on the other hand, is what you actually see when you look at something. Okay, it's defined based on physical appearance. It's what's actually expressed. We'll talk in the next chapter about genetic disorders in humans, and those are also considered phenotypes. So even though you can't necessarily see that, there are symptoms and other types of things that come along with that, and we still consider that a phenotype. Okay. So if we think about this statement here, what we know about genotypes and phenotypes based on what we just talked about is that any scenario where I have a dominant phenotype, remember, I don't know if I'm going to have a heterozygous genotype, big G, little g, like these two, or if I could possibly have a homozygous dominant genotype, big G, big G. So two organisms can actually share the same phenotype. These guys can all be green, but actually have different genotypes in this case. All right, so it's not always going to be so cut and dry. Again, the only exception to that is this one with the recessive trait. Okay, there's really only one option for that one. Okay, so moving on to Punnett squares, and I just have a few slides left in this section of notes, and then we'll do the Punnett square screencast. So Punnett square is a grid system for predicting possible genotypes and then phenotypes resulting from a cross. Punnett squares, it's capitalized, the word Punnett, because it's named after the man that created it. His name was Sir Reginald Punnett, which is kind of a weird and goofy name, but that's where it came from. And so essentially what we're doing is we're trying to represent the likelihood of inheriting specific traits. So on every Punnett square that you draw or that you complete, the axes represent possible gametes for each parent. So if we look at this one, the axes are here, either on the top or on the left. And for any Punnett square, we only ever put them on the top and on the left. Nothing will go on the right, and nothing will ever go on the bottom. Okay. The interior boxes are going to show us exactly what we get in our offspring. So this is parent 1, and they're representing them in green. Parent 2 here in purple. And you can see how we're combining these A alleles, to create genotypes in the offspring. And each offspring, notice, has 2 alleles, just like each parent started with 2 alleles, because each parent inherited those 2 alleles from their parents, right? So to have a complete genotype, to have a complete organism, all organisms, and this is independent of whatever type of species they belong to, but you've got to have 2 copies of these things if we're talking about sexual reproduction, okay? So when we look at Punnett squares, we can infer phenotype. We can directly read off genotype. Like I can say this organism here is big A, they are homozygous dominant. I can look at this one here and say this is big A, little a, or heterozygous. Same for this organism here, and same for this one right here, little a, little a. What you should know, though, about this, what you need to kind of keep in mind, these are not actual offspring at this point. So this is simply predictive. All right, we're saying that if these 2 parents mated, they have a 1 in 4 or a 25% chance of producing offspring with the recessive phenotype, little a genotype. We also have a 1 in 4 chance of creating offspring here that are homozygous dominant and have the big A genotype and also the dominant phenotype. However, if you consider just phenotype by itself, right? I have this one here, big A, this one down here, big A, little a, and this one over here, also heterozygous, big A, little a, all of which have at least one big A. So in reality, what that tells me for phenotype is that all 3 of these would express that dominant trait, whatever that is. So I would actually have a 3 of 4 chance of producing an organism with the dominant phenotype. So that's 75% chance of my offspring that would actually be dominant. Okay. So the small crosses like the one you just saw and the one on this screen are called monohybrid. And mono, if you remember from Latin terms at the start of the year, means single. So we're looking at a single trait. All right. In this example, it's flower color. All right. And we, again, have copies. We still have 2 alleles because we're talking about coming from mom and dad, right? But we're looking at only one trait, flower color. So we have 2 different parents. We cross them and we can see, the resulting offspring. The difference here now, we have an additional type of cross, and I'm going to walk you through how to do this one too. It's much more complicated. But this is called a dihybrid cross, and this is looking at the inheritance of 2 traits, okay, or 4 alleles, because now we're looking at 2 different genes, each of which mom and dad each donated 1 copy per gene, okay? But you have to consider all possible options. So we have to include all 4 alleles. So you can see how much larger this is. It's 4 times larger than the other one. It's basically 4 monohybrids. But notice how my axes are different, right? I have Ys and Rs and not just one single letter, because in this scenario, we're looking at both P color and P shape. So yellow versus green, round versus wrinkle. And so this becomes much more complicated and much more difficult to try and predict. But based on Mendel's stuff, we can still make some accurate and come up with some accurate probabilities about how often we see these different phenotypes. Because again, these things do separate independently. They don't blend. It's all independent. Okay. So speaking of that, Mendel had another law. We talked about one of the laws with section 12.1, I believe, and that was the law of segregation. This one is called the law of independent assortment, and it's a second and final law. According to Mendel, this means that genes for different traits can segregate independently during gamete formation. So in other words, you can have peas here that are yellow and round. You could also have peas that are yellow and wrinkled. You can have peas that are green and round and green and wrinkled. It is not just one and the other all the time, right? You don't have to have both red, excuse me. You don't have to have both yellow and round at the exact same time. There are other possibilities there because these are found on different chromosomes, they're different genes, and they're inherited independently of one another, okay? So that's all for 12.2. I'm going to go ahead and do the screencast. So if that is part of your assignment today, make sure you go in and watch that for the Punnett squares. Other than that, if you need anything from me, let me know. Otherwise, I will see you for 12.3 in a little while. Bye, y'all.